Good morning. It's April 21st, and this is your daily brief in science. Here's everything you need to know. In a White House Oval Office ceremony, the president signed an executive order directing federal agencies to speed up reviews of certain psychedelic drugs, including ibogaine, to accelerate medical research and potential treatments for serious mental illness. Supporters, including veterans advocates like Marcus Luttrell, see potential life-saving impacts, while medical experts stress the need for robust clinical evidence and safety data. The report notes ibogaine's traditional use and cardiovascular risks, and it emphasizes that no psychedelic has FDA approval yet, while noting state experiments with psychedelic-assisted therapy in Oregon and Colorado. The piece also includes a newsroom disclosure that Barchart Insights did not hold positions in the securities mentioned. It places the development in a broader trend of political leaders and media figures shaping public policy discussions, contrasted by Joe Rogan's influence and rapprochement with Trump against Tucker Carlson's ongoing criticisms. The Associated Press contributed reporting for the article, and user comments accompany the piece, with critics calling the Oval Office moment insecure, while others discuss clapping and the broader political discourse. Healthcare cost and affordability context are noted as part of the policy discussion around access to psychedelic therapies. Sources cited include White House actions, media coverage, and responses from Compass Pathways, highlighting cross-cutting interest from government, media, and industry. Market reaction showed a notable stock dip and increased trading volume following the news, with Compass Pathways and related biotech peers mentioned in post-announcement chatter. A new approach in the fight against solid tumors is on the radar, as Verismo Therapeutics reports early Phase 1 data for SynKIR-110, a KIR-crown-AR T-cell therapy designed to resist T-cell exhaustion in tough tumor environments. The therapy uses a multi-chain NK-inspired architecture that activates only when it encounters its target, potentially delaying exhaustion in the tumor microenvironment. In AACR 2026 data presented at STAR-101, SynKIR-110 showed activity in advanced mesothelin expressing solid tumors, including ovarian cancer, mesothelioma, and cholangiocarcinoma. The trial is still escalating doses to establish the recommended Phase 2 dose and to further assess safety and preliminary efficacy. Expansion cohorts are ongoing to confirm safety and look for signs of efficacy at the selected dose level, with enrollment remaining active as researchers monitor safety. Interim results include a partial response at dose level 3 per RECIST, with the response continuing beyond 3 months as of the September 2025 cutoff. In broader results, one patient at the highest dose achieved a partial response, while others showed disease stabilization at dose level 3. Dr. Janos L. Tanyi of Penn presented the data, noting that Penn intellectual property is licensed to Verismo, underscoring a Penn origin-aided collaboration. 9 patients across 3 dose cohorts demonstrated a favorable safety profile with no dose-limiting toxicities, low-grade CRS in a subset, and no ICANS. The press release carries forward-looking statements and includes standard licensing and investor media contact details. A new study in Current Biology finds cocaine contamination in freshwater alters Atlantic salmon behavior in the wild, with hatchlings swimming farther and dispersing more widely than unexposed peers. Researchers implanted slow-release devices to deliver environmentally relevant doses and tracked salmon with acoustic transmitters for 2 months, comparing them to drug-free controls. Benzoylecithin, a cocaine metabolite, produced a stronger effect than cocaine itself, highlighting the importance of including metabolites in environmental risk assessments since they're often found at higher water concentrations. The study suggests current risk assessments may miss important contaminants by not accounting for drug derivatives. Movement changes from exposure can influence feeding, predation, and population structure, signaling potential ecosystem-wide implications. Experts call for better wastewater management and greener medicines to reduce pharmaceutical pollutants and raw sewage entering waterways. The research emphasizes the need to study wild populations beyond hatchery-reared fish to understand real-world effects. Griffith University scientist Dr. Marcus Michelangeli notes, pollution is a growing concern for aquatic ecosystems with wildlife routinely exposed to human-derived drugs in waterways. Altered movement patterns could cascade to ecosystem dynamics, affecting feeding, predation risk, and population structure. While cocaine exposure did not pose a human food risk, the findings raise broader concerns about pollutants entering ecosystems via wastewater that isn't fully treated. The study argues for higher resolution tracking and broader cross-species studies to capture the ecological risks as illicit drugs increasingly appear in waterways. Overall, the findings point to cascading ecological risks from cocaine and metabolite pollution and stress the need for risk assessments that include metabolites, not just the parent compound. A real-time milestone in black hole physics emerges from Cygnus-X1 as researchers measure its jets on the fly, not just from averages over years. Using a network of Earth-based radio telescopes, they tracked instantaneous jet power and speed, a first for this system, and a major step in accretion physics. The jets travel about half the speed of light, delivering energy roughly equal to 10,000 suns in luminosity. The key insight ties 10% of the matter falling into the black hole to the jet output, aligning with long-standing theories about how black holes channel energy. The jet energy was inferred from how the jets interact with the wind from Cygnus-X1's supergiant companion, which deflects the jets and provides measurable cues. By accounting for this deflection and using archival data, the team could determine jet strength at the moment of emission. The result establishes a practical benchmark for black hole feedback, useful for interpreting jets in more distant galaxies and in supermassive black holes. Published in Nature Astronomy, the study highlights calibration opportunities for simulations across black holes of different masses and scales. The findings also set the stage for future observations with next-generation radio facilities like the Square Kilometre Array. The collaboration, led by Curtin University's CIRA and ICRAR, includes researchers from Oxford and multiple universities, reflecting strong international cooperation. Looking ahead, the methods could guide SKA-era observations, improving understanding of how jets drive shocks, turbulence, and structural changes in the universe. A new stem cell model from the University of Michigan recreates early human development in a way that forms yolk sac-like structures from a single starting population without direct genetic manipulation. Using circular micropatterns and BMP4 signaling, researchers guided epiblast-like cells into a 3-layer, gastrulation-like disc, with an amnion-like cavity on top and a yolk sac-like region beneath. The model reaches peak embryo similarity around day 8 in culture, roughly 16 to 21 days post-fertilization, with about 15 to 20% of cultures forming yolk sac-like structures depending on the cell line. Yolk sac identity was validated through lineage tracing and activation of the HNF4A gene, a marker associated with yolk sac and visceral endoderm, and cross-validated against post-implantation monkey embryo data from collaborating groups. The work was conducted at the University of Michigan and corroborated by Chinese Academy of Sciences researchers using non-human primate data. The model consists solely of epiblast-like pluripotent stem cells confined in a 0.8 mm circular disc, which self-organizes under BMP4, signaling into a trilaminar disc with ectoderm, mesoderm, and endoderm analogs plus an amniotic cavity. Geometric confinement and advanced microscopy, genomics, histology, and flow cytometry supported the multidisciplinary engineering biology approach. The yolk sac-like structure arose within a gastrulation-focused framework, modeling early embryo organization with an amnion-like cavity on top and yolk sac-like region on the opposite side. Limitations include the absence of trophoblast or placental tissue, thicker than normal body layers, and eventual loss of embryo-like organization, with development constrained by 14-day culture rules until further cues are added. The study highlights the potential of stem cell models to illuminate early development and pregnancy loss while noting ethical and regulatory considerations as cultures progress beyond traditional 14-day limits. Notable advances include observing embryo-like structures at roughly 16-21 days post-fertilization in a transgene-free model, bypassing many ethical and technical barriers associated with natural embryos. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.